All right. So good afternoon, everyone. So my name is Olaitan. I am the training officer for the African Society for Bioinformatics and Computational Biology. So that's ASBCB for short. The aim of ASBCB is to advance the knowledge of bioinformatics, primarily in the continent of Africa. So as part of me being the training officer, I do organize workshops, webinars, and hackathons with the aim of increasing the representation of life scientists that work from Africa and also from the diaspora. And it's mostly virtual events so far that have been done, and some of the impact and output from those you will be hearing about in the next few minutes. Okay, so when the event started, it used to be hard to actually find a lot of people attending the events. So we would solicit for ideas around what people would like to learn or do in these workshops and hackathons. And I remember those days I would be very intentional about just trying to find at least a very few number of maybe interested applicants. But fast forward about a couple years after that, the application pool actually really exploded. You know, it got to the point where from always being intentional about ensuring that, you know, a good number of people apply to where we have to kind of have a large wait list to be able to accommodate at least a decent number of people that can learn from the events. When it started, we probably were in maybe 18 or so countries, which mostly were Africa. Then over the course of years after that, we got to beyond Africa. So like you can see, we have African countries, maybe like 27, 28 right now, South Africa, Kenya, and the likes, all across, Northeast, Southwest. But then we also have from other places like Canada, the UK, the US, Mexico, Australia, Belgium, Spain, Ireland, and the likes. And a lot of these applicants that attend eventually, the ones that are shortlisted, they work, even though some of them are Africans, some of them are non-Africans now, they work from all these places. So that's decent success. And the growth has been quite organic. So the research standard we follow in these events is generally open science, right? So I tell myself that I'm the police for the workshops and the hackathons. Enforce fair standards, findable, accessible, interoperable, and reusable data, code, and pipelines. Code has to go on GitHub. We do write manuscripts also from some of the projects that we do, and it has to have a DOI. So people are collaborating, working on Google Docs and things like that from different locations. And the attendees like the idea of meeting new people at the events where they can actually collaborate and do cool stuff, fun science. So open access, when we do submit a manuscript and the journal then says, oh, you have to pay the APC now that it's accepted. And even if we don't have the APC, and the attendees of this event are saying, oh, let's just do the paywall. I said, no, we're not doing any paywall. So I'm that strict with making sure that it's all open science and open access. And the goal is, of course, to improve the quality and the efficacy of science. Because it's mostly virtual, sometimes we do have events like conferences, like last month, for instance, we had this ASBCB conference in Cape Town. That became a platform to connect physically with some of the people that do attend the event. So we're taking pictures. So we took a picture just in front of the banner for the ISCB, who basically partners with the SBCB to run that conference. And the people you have there like the 6 people there are all, the 5 other people apart from me, of course, are all people that have attended the codeathons, whether the last one that we did or sometime in the past. And then also during the networking event, I ran into somebody else who had joined, I think, the April codeathon last year. So I took a picture. And then the picture you have on the right is a poster presentation by one of the hackathon participants presenting a work on clinical node generation to help clinicians using LLMs. We have some great impactful work done in these workshops and events. So the way it works is that some of the workshops I'm teaching, so I teach Nextflow programming in the workshop. It's not like the super extremely advanced programming level thing, but it's still basically you're able to do the hello world. It includes things like understanding of channels, processes, doing a typical RNA-seq pipeline in Nextflow, working on the pipeline by yourself, building de novo. And then I also still get into configuration, so you could run things from places like NF Core and the likes. So I would spin up cloud instances, GCP instances, to have people work on these things. But using things like code spaces now should make the work easier and, of course, more affordable. So this is a tool paper we published earlier this year. It's XVAR. So this is not Nextflow, this is R. So it's an R package. It does differential gene expression analysis. You can also do your calling of SNPs, calling of indels, and copy number variants. And it has 3 functions for data visualization of looking at copy number variants, single nucleotide polymorphisms, as well as indels. And that's in scientific reports. It's out there to read it. We work on 8 model organisms: human, mouse, C. elegans, yeast, and I think 4 more species. Okay, so this is another paper that we published. So this is ProStruct. So it's a Python package. It's open source as well to do 3D structure prediction using homology modeling. It's in Frontiers in Chemistry. It's Python, and we do have a Docker container for it also in the Docker registry that could be used if you want to run the 3D prediction by yourself, instead of using the web server component that we also built, where you could upload your template and then do your fastest sequence and then do the structure prediction. Then we have NeuroVar. So that's another tool that we published last year. So NeuroVar basically is open source as well. It does visualization of gene expression, SNPs, and indels. And you can look at various things, volcano plots, adjust your p-value, log fold change, and stuff like that. So yeah, that's also out there. You can read it in Gigabyte, published. Okay, so this is a Nextflow summit. So let's get to more Nextflow, Nextflow things. So I'll be talking more about these other dashboards that we developed, and we do have the manuscript right now under review in scientific reports. I did get word from the editor, I think yesterday actually, so I was resubmitting the second round. So I think this is moving forward. So what does it do? So Rare Insight. So Nextflow enabled rare disease report generator for clinicians and patients. Now, the idea of this is to look at people that are mostly working from low middle income countries, underserved places that might have genetic disorders, but then they have a hard time accessing dashboards. Actually, even getting the diagnosis is even hard. There's a lot of misinterpretation, very low literacy in those places. So the idea is to actually have a dashboard that can help in communicating rare disease, genetic information to rare disease patients. So I have some statistics there. Over 6,000 different rare diseases exist right now, 1 in 2,000 people. I know it's more like population-specific definition for what a rare disease is, but at least in South Africa, 1 in 2,000 people having a disease, you would say it's a rare disease, right? 72% of the rare diseases are genetic, and globally, 30 million people are living with rare disease. For the most part, there is no known cure for a lot of these diseases. So there is this diagnosis or DC for rare diseases where a patient goes to the clinic to get diagnosis. There is either misdiagnosis or just complication in knowing what's going on, back and forth and back and forth. And at some point, there is the possibility of actually being able to know what the disease is, but then it gets to the point of, what do we do with this knowledge or this information? We have available dashboards right now that do reporting on rare diseases. I have some of them there, SOPHIA, DDM, VARSIC. Generally, these are mostly proprietary. So there's not a lot of people that can access them, especially from low middle income countries, right? So there are challenges with being able to have the access. There are concerns sometimes around data privacy and security and things like that. So our idea was to build a rare disease reporting dashboard that will be, easy to access and more understandable for both non-specialist clinicians as well as caregivers and even the patients. So we did obtain data from the European Nucleotide Archive to build a dashboard that had a rare disease that we could use in testing the dashboard that we built. So it's powered by the NF Core rare disease pipeline. Basically, the input to that is it could take whole genome sequencing data, it could take whole exome sequencing data, but the one we built, we use whole exome sequencing data to do the development. And you can also put targeted genes also as input FASTQs. It produces VCFs. So the VCF output from the rare disease pipeline that we import into our dashboard. Other options are things like GATK. SAREC is mostly cancer-based rare disease, variant calling pipeline. Okay, so what did we do? We installed our RStudio with the necessary packages and designed the user interface functions as well as the server functions and did the deployment, built all the necessary code to do the reporting and then deployed the dashboard. So this is just the NF Core rare disease pipeline. Okay. What was the actual goal or what we hoped to contribute to the field when we started to work on this is to improve the genomic literacy through tutorials, as you will see shortly in the dashboard, make it open source, so it's collaborative and not just limited to only people that have funds to be able to access the dashboard, and access to resources for patients, caregivers, and people that have little knowledge of genetics, basically. So it's patient-centered. So the limitations are that the dashboard is not suitable for novel or unreported variants, so it's only as good as what ClinVar knows. So the best annotation in ClinVar is what the dashboard will rely on, and we are not yet able to export results to a clinical report because of just being able to parse certain fields from the VCF right now. So in the future, we hope to be able to achieve basically these things that I just talked about and solve them, like filtering and searching the VCF, through the VCF file, allow multiple input types into the dashboard, deploy the dashboard to a live web server and not just on your RShiny app locally, and things like that. So here's the team that did this development, just to show the representation situation that I was trying to talk about earlier. So we have Kim from South Africa, we have Firas from Tunisia, we have Eva from Uganda, and we have Gideon from Ghana. So that's good. I think we're making progress there. Okay, so this is what the dashboard looks like. So you have your home tab, and you have the tab for understanding your genetics and the services, as well as the acknowledgements. So the home tab just tells you what Rare Insight is about. Understanding your genetics just gives you more information about genes, what they are, DNA, and some basic tutorial, if you will. The services is how you actually visualize what the VCF has, the information in the VCF. So the user can input some information here and just visualize, based on whatever VCF you uploaded, visualize the table in the VCF. You can actually visualize here and also look at the quality of the data. So you have your search panel to search for terms, gene names, variant name, disease name, and things like that. So this is the tabular format and then the plots format for your looking at the VCFs. And this is just more like the next tab, just more like information for the patient, additional information and links for the patient. This is the workflow. And yeah, there are people that support the work and help with organizing the events and things like that, like Joe Johnson, Shivani, George Chenemi, Kim, who actually led this Rare Insight project, has also been very supportive in getting the workshops and the codeathons organized. And people from like the Biodata Sage, we have NIH ODSS support, the EBI, and some other colleagues like Derek, Justin, and Hamna. So that's my email in case you have any questions. Thank you very much.
Thank you so much for sharing this with us. I think it's really important to see. I think we have a responsibility to make sure that the things that we develop are usable by everyone in the world as genomics improves, as genomic medicine improves, that everybody gets to benefit from that. And it's great especially to see efforts at community building within the countries so that they can serve themselves, and that's wonderful. So we have a few questions. Marcel is asking, do you plan to create an NF Core module for XVAR? It's a great question.
Okay, I'll take that up. Well, actually, before this presentation, no. But then, yeah, it's a good consideration, for sure. Because RNA var does variant calling. The input is, RNA data, right? But XVAR does, input is also RNA data, but then it does differential gene expression, visualization, and analysis. It does the actual variant calling, SNPs, and does CNV. So yeah, it would be good to have it in. And of course, so thanks for that suggestion. It's work, though.
Was there a comment in? Oh, okay. Also, well, Maxime was right over there. Is asking what would you need to have SARIC more compatible within your framework?
Oh, to have SARIC more compatible?
Any feature requests?
Yeah. Well, okay. Feature requests. I think SARIC is just targeting a different class of disorders than the NF Core rare disease. So. Yeah, I think leave SARIC where SARIC is.
I like that answer.
Perfect answer.
Okay. Any comments or questions in the room? Anyone? No? Oh, yeah, Ken?
I wanted to know about how you do the virtual hackathons and what you do to make them a good experience for attendees, because for things that we've run in the past, we know that can be particularly challenging to make sure people feel like they're engaged and have a good experience in these virtual events.
Yeah, that's a very, very good question for sure. Having online events, driving engagement could be quite challenging. So one of the things we do is we have meetings. So the attendees, so we schedule meetings. So there's I would say, workshop level or codeathon level meeting for all attendees. And then we put the control of the individual team meetings to the team leads and say that please work with your team and try as much as possible to engage with them and have meetings where you're discussing things that you want to put into whatever projects you're working on and what you're learning and things. So that helps because we have a way to report what's happening even within team to the rest of the codeathon in terms of the facilitation. So it's like two-tier now. So all participants attend the workshop and codeathon level meeting, and then within team, they have their own meetings. So I lead the entire event, but then I have a few people that kind of assist in overseeing what's happening in groups or teams. So they are the ones mostly that actually interact with the teams. So the engagement kind of happens automatically because of the way it's structured. But the idea is that the teams should meet and then get to the facilitators to tell them what they're doing or if they have challenges. Then I make myself available to meet with everyone and then just have people report and talk about what they're learning or what they're planning to do with their work. So yes. But being fully online, we do have, I would not say that we have 100% engagement. So the motivation is always high in the beginning of any event, like normal. But then by the end of the event, we don't have that. Let's say it was 90% at the beginning. Of course, it won't be 90% engagement at the end, but it's still decent. At least we have those outputs to show that, yeah, things are happening.
And it does feel like we could learn from that a little bit in terms of, as we grow the, the NF Core hackathons, the NF Core distributed hackathons that we do yearly in March. That's also a challenge, having remote sites and remote participants engage. And on that topic, I wanted to ask you if you see an interest in having activities joined together, like having maybe sites or groups within your scope join the NF Core hackathons, like would that be useful? And if so, what could we do to help make that happen?
Yes, I think it would be useful to just increase the literacy of Nextflow among attendees and challenge the attendees more. And yeah, what needs to be done is just, kind of discuss with you and let's, plan things out.
Fantastic.
Sure.
Sorry, I guess you don't need that. But yes, we have a couple more questions from on Slack. I'll take one more. What are the unique challenges that are faced when doing bioinformatics in the continent of Africa, within African countries? Are there any specific challenges that you face?
Yeah, of course, there's a number of challenges like infrastructure. We are not as rich, so access to funds, access to actual compute. Of course, we spin up cloud instances for some of these analysis, but then some people have internet access challenges even in their place. So they're dropping off sometimes on the Zoom calls, for instance, right? Or, you're connected to some GCP instance or AWS instance, but then your link keeps on dropping back and forth. You have to background the process to make sure it's running at least, so even if you get off the terminal, you can still get back in. So all those things are there. We don't have that much money, so people are barely just trying to survive. And then, yeah, if there could be more money put into research and developing people from Africa, there's a lot of talent in Africa. I am African and I was born and raised there, right? So I just have that passion to use my experience and all the things I've learned and exposure, just give back, and because there's a lot of talent in Africa, well-motivated individuals, but very little access to actual opportunities and funds.
Well, it sounds if we have any friendly cloud providers in the room who want to provide some credits. I don't know. Is Angel still here? Not to point anywhere. Anyway, we'll try to help in as much as we can. Thank you so much, Olaitan, for sharing. Thank you.